For decades, nephrology viewed the transition from acute kidney injury to chronic fibrosis primarily as a structural failure, a gradual accumulation of scar tissue that physically compromises the kidney's filtration system. More recently, researchers have shifted their focus from this visible scarring to the microscopic processes occurring earlier in the disease cycle, specifically the state of genomic maintenance within the cell itself. Data from genome-wide association studies has identified a specific genetic target at the center of this process, the epigenetic regulator known as TET2. Parallel analysis using single-nuclear RNA sequencing reveals a second related target, the DNA repair gene INO80. Today, we will map the molecular pathway that connects these two distinct genes into a single cohesive mechanism. We will explore how TET2 acts as a guardian of the proximal tubule, chemically modifying the DNA to allow INO80 to repair double-strand breaks. Then, we will track how a breakdown in this specific repair system provides the trigger for the body's innate immune response. This sequence suggests that chronic renal fibrosis is not a standalone inflammatory event, but the result of a specific breakdown in epigenetic and DNA repair mechanisms. To trace this mechanism, we begin inside the nucleus of a healthy proximal tubule epithelial cell, operating under normal physiological conditions. Here, the TET2 enzyme acts as a methylcytosine dioxygenase. Its role is to chemically modify DNA to regulate which genes the cell can actually access. To catalyze these reactions, TET2 requires a specific supply of cofactors, alpha-ketoglutarate, iron, and vitamin C. When fueled, the enzyme targets a chemical tag on the DNA called 5-methylcytosine. The enzyme oxidizes this tag, converting it into 5-hydroxymethylcytosine. This chemical shift alters the DNA's physical properties, allowing tightly wound chromatin to relax and expose the underlying sequence. In the proximal tubule, this unwinding occurs at the INO80 promoter region, allowing the cell to continuously transcribe instructions for the INO80 protein. INO80 functions as an essential ATPase subunit within the cell's chromatin remodeling complex. This complex is a primary driver of the homologous recombination pathway, the mechanism cells use to repair severe double-strand breaks in their DNA. In these cells, the capacity to respond to DNA damage is tied to this initial state of chromatin accessibility. This system is often disrupted during acute kidney injury, whether through exposure to toxins like cisplatin or the physical stress of a blockage. Genetic risk variants, or the natural processes of aging, can also act as the initial point of failure. In response to these insults, the cell rapidly downregulates its production of TET2. This chart shows statistical associations across thousands of patients. The data highlights a clear vulnerability. The peak risk variants for chronic kidney disease map directly to mutations that lower the expression of TET2 and INO80. Inside the cell, the loss of TET2 means the INO80 promoter is no longer modified. It begins to remethylate. This remethylation causes the chromatin to wind tightly shut, silencing the expression of INO80. Without INO80, homologous recombination fails. DNA breaks begin to accumulate, and the cell loses its ability to repair them. The consequences become physical when the cell attempts to divide. The damaged chromosomes fail to segregate properly during mitosis. As the nuclear envelopes reassemble in the two new daughter cells, an orphaned fragment of DNA is left behind, excluded from the primary nucleus. This isolated, misplaced fragment is known as a cytosolic micronucleus. To the cell's internal surveillance systems, the presence of DNA in the cytoplasm acts as a primary trigger for an immune response. This trigger is recognized by an innate immune sensor called cGAS, which patrols the cytosol. When the cGAS enzyme encounters the cytosolic micronucleus, it physically binds to the DNA, interpreting the host's own genetic material as a foreign threat. This binding initiates the synthesis of a messenger molecule called cGAMP. The cGAMP molecules diffuse through the cell until they reach the endoplasmic reticulum, where they bind to a receptor protein called STING. This binding activates the STING pathway. The result is a sudden release of pro-inflammatory cytokines, including IL-1 beta and TNF-alpha. These signals move into the surrounding tissue, recruiting macrophages and initiating a state of chronic inflammation. In this model, the cGAS-STING cascade is the biological bridge that translates a failure in DNA repair into irreversible tissue fibrosis.
We can now view the entire progression as a single continuous chain, the TET2, INO80, CGAS, STING axis. Mapping this sequence allows researchers to identify specific points where this cascade can be intercepted. This diagram shows the logic of current research into potential therapies. One approach focuses on the upstream epigenetic state. Compounds that activate the AMPK pathway are being established their ability to stabilize the TET2 enzyme, preventing its degradation during injury. At the same time, maintaining the levels of metabolic cofactors like alpha-ketoglutarate and vitamin C may help the cell maintain its INO80 repair complexes. For contexts where DNA damage has already occurred, the focus shifts further downstream. Researchers are investigating STING inhibitors to block the inflammatory response even after micronuclei have physically formed in the cytoplasm. The preservation of kidney function is closely linked to epigenetic health. Protecting these DNA repair complexes offers a pathway to slow or stop the progression toward tubulointerstitial fibrosis.